So now it's my pleasure to introduce Radu Botgros from the EMA, who's going to speak to us about the EMA position on antifungals, which was quite divergent some years ago, but I think has come together a little bit more than now.
Yeah, let's hope that. Thank you very much, Professor. And good morning to you all. Thank you for coming here this morning. I'm going to talk to you about the European Union regulatory framework for development of new antifungal medicines. And this is the history of guidance starts back in 2003, where there has been a note of guidance developed for products for antifungals that were aimed at treating and prophylaxing invasive fungal disease. This was done in response to repeated applications or scientific advice proposals that had been received by the CHMP, by our main scientific committee at the EMA, to seek approval based on non-randomized studies with or without external controls, often in patients who had failed or were intolerant of initial antifungal treatment. And then between 2003 and 2010, there had been a number of developments in terms of approvals and scientific advice application received, which led to a further revision. And the revision of 2010 is actually still in force. So the guideline from 2010 is still in force. We are looking into updating that as soon as possible. We actually had that in our plan, but of course, due to the change of the seat of the agency recently, this has been put on hold, actually. Just to mention that the guidance, of course, reflects the EORTC recommendations and categorization of fungal disease. So just a few salient points for you out of this guideline. To start with, when it comes to dose regimens, the guidance states that the selection should be based on all available non-clinical data, human PK data, and the exploration of PK-PD relationship for selecting regimens to be studied in confirmatory trials. We have, in 2015, developed a separate PK-PD guidance for developers, which goes into antibacterials, antifungals, and antimicrobial agents. And while we acknowledge that the experience in using PK-PD for antifungals may be a bit behind antibacterials, this is another guidance that I would like to point you to when constructing the program. When it comes to invasive aspergillosis, which is one of the main diseases and the topic today, the guidance states that the recommendation is to have a prospective randomized active control trial. We have, and CHMP has accepted in its recent approval, single pivotal trials for each indication. And as Sumari has stated, isavuconazole is the latest one, which has had one pivotal trial per each of the two indications approved, one of them being invasive aspergillosis. The primary analysis should be conducted in patients confirmed to have proven or probable disease. And when it comes to the comparative agent, the preference is to have one single comparator, but if it's not possible, the choices should at least be restricted and should be pre-specified in the protocol. It is recommended to use an independent adjudication committee which is blinded to treatment assignment to determine eligibility and outcomes. When it comes to the presence of fungemia, it is very important to make all efforts to identify the primary foci, because of course the removal of a suspected catheter or suspected catheter source may not always be enough to actually make sure that the fungemia is because the need is that fungemia is persistent and that we know as much as possible the primary foci so that this can be counted in primary analysis to support the indication. One of the differences, the main differences between US and the EU is the primary endpoint. Our preferred primary endpoint is clinical. So it's global clinical responses test of cure. But I think it's important to mention that the CHMP actually has accepted predefined primary efficacy endpoint of all-cause mortality at 842 and 84, provided that both endpoints go in the same way. So the global clinical response rate were supportive. When it comes to the non-inferiority margin, well, actually, it's rather difficult to justify the margin, but in general, a 10% non-inferiority margin has the one that has been accepted. A few words about rare invasive fungal diseases. And I should start by saying that there have been some proposals of sponsors conducting non-randomized studies in patients with such infections. Some were accompanied by historical controls as comparators. There were some proposals based on such studies with some support from PK/PD to support the dose selection. In front of these proposals, actually, the CHMP advised that at least one randomized controlled trial in an invasive fungal disease, be it Candida or aspergillosis, should be conducted before or in parallel with the rare pathogen study. And of course, the dose can be justified using the efficacy results from the RCT in invasive fungal diseases plus the PK/PD analysis using the human PK data. It is important, therefore, to highlight that at present the guidance does not foresee that the treatment of rare pathogens could be based only on RCT in Candida or Aspergillus plus PK/PD. So there is a need for a study in rare invasive fungal diseases. The preference is that a randomization step is used, and in view of the low numbers, unbalanced randomization can be used. There is a preference of having separate studies by fungal type. The comparator should be either a licensed agent, if existing, or if not, a best available therapy. Of course, if there is nothing approved or there is no treatment that is considered adequate, it could be appropriate to seek to demonstrate superiority of the test regimen versus best available. As a general rule, studies in refractory invasive fungal diseases, which is yet another important topic, should be conducted only after efficacy has been shown for the drug in one or more specific types of invasive fungal disease. The patients should have proven invasive disease, except for aspergillosis, in which we also accept probable cases. And those have to persist or have progressed despite previous antifungal therapy. It's important to avoid those patients, to enroll those patients in which discontinuation of the therapy has occurred due to intolerance to treatment only. The primary objective is in need of discussion depending on whether or not an active control design is possible. Just a few words about prophylaxis. So here the recommendation of the guidance is the study should be conducted in prophylaxis only after the agent has shown satisfactory efficacy in the treatment of invasive aspergillosis. In that case, a randomized controlled trial versus a comparative regimen will be needed. The discussion about the non-inferiority margin needs to take place ideally in the context of the scientific advice of the CHMP, because the guidance does not specify the magnitude of the non-inferiority margin. And then the indication that could be granted if the results of such study are convincing is likely to reflect the evidence for prevention due to specific fungal types. Just a few words about the use of biomarkers and surrogate endpoints. Some of them overlap with what Sumati just said, so I won't be leading you through all of this, but indeed some biomarkers may act as surrogate endpoints, not all of them. They are very important because they can be used to shorten or to optimize drug development. But what is important, what is pivotal actually, is the context of use of such biomarker, whether it is used for enrolling patients or for the final outcome, for the main outcome, it makes quite of a difference. So the main question is a biomarker intended as a surrogate endpoint for clinical efficacy or not? While answering as a developer to this question, it's important to define the populations for which such biomarker could be used, the phase of the clinical trial in which the marker is supposed to use, as well as the importance of it. Is it a primary endpoint, a secondary endpoint? And so the level of evidence required to support the intended use of the biomarker depends then on that context of use. The level of evidence that regulators will require would be highest if such an endpoint is intended as a primary endpoint in a pivotal clinical trial aimed at full approval. At the time of the marketing authorization application, depending on that evidence, we can grant either a full marketing authorization or a conditional one. It is important also for you maybe to know that we have a qualification process of biomarkers at the EMA, which means essentially that that biomarker gains regulatory acceptance to be used. We have two types of opinions or outcomes. One is the qualification advice, in which these contexts of use, the objectives of the studies themselves, the methods, the protocols are submitted for assessment, and then the CHMP provides an advice letter with an opinion on that proposed plan. And then at a later stage point where some additional data have been generated to support so the biomarker or the surrogate endpoint, then there could be a qualification opinion. It's an EMA opinion which actually qualifies the biomarker for the intended context of use. And just for your reference, here is the link to which has more details to this particular process. So in summary, as I said, the guidance is a 10-year-old guidance. It is still current. We are looking at updating it as soon as we can. We have had a few new approvals since 2010, relatively few requests for scientific advice regarding treatment and prevention of invasive aspergillosis. We have, and we recognize that there have been quite important developments in the field of PKPD in the last 10 years for antifungals. There is quite some flexibility, or there has been quite some flexibility from the CHMP when it comes to the primary endpoint that has been accepted for the antifungals, for the few antifungals that have been approved recently. For rare pathogens, it is important to first establish efficacy in Candida or Aspergillus, and then use those data to support results of the smaller randomized trials in the rare pathogens and the PKPD to support the dose. And last but not least, the prophylaxis should be assessed after treatment. So thank you very much.
Great.